actually going to have Yakir speaking up next, and he's going to be talking about powerful and accurate case control analysis of spatially resolved molecular data. So thank you for the flexibility. Thanks, Yakir, and let's welcome him up.
All right. Thanks. So I'm a computer scientist and a physician who sees patients with autoimmune disease. I work in the Raychaudhuri lab here in Boston. And I want to tell you about a method we've developed for finding disease associations in spatial data. As has been already described, spatial data are advancing very fast. We can now profile really quite large numbers of proteins and genes at very high spatial resolution. And as you've already seen, this has been a really rich area for methods development, and there are some really sophisticated approaches now for analyzing spatial data, many of which use advanced ML approaches. And thanks to these methods, such as what Dana just showed you, we can now annotate tissue at really, it parse out tissue structure really in quite granular detail. Today I want to talk specifically about finding disease-associated spatial structures, and I want to do this in a statistical framework. Given a spatial data set with many case samples and many control samples, are there patterns that are statistically significantly different between cases and controls after multiple testing correction? And while it might seem like our ability to successfully parse out tissue structures in spatial data should translate intuitively into well-powered statistical case control analysis, we found that these are actually two quite distinct problems. There's issues like batch effects and finding flexible representations at the sample level that allow samples to be compared to each other. These are crucial for case control analysis. And so even though some existing methods have been used in a case control setting, it wasn't clear to us how effective we should expect them to be in general. To look at this, we used several existing tissue annotation methods to analyze a spatial transcriptomics dataset of 75 brain samples from 27 donors in a dementia cohort. Now, doing benchmarking of this type is challenging. Not all the methods necessarily output p-values. Some of them don't even allow multiple samples as input. So what we did was we chose 5 methods that were efficiently runnable on a data set of this modality and sample size, and we used each of them in this standard way. So for each method, we annotated all the samples at the level of small spatial neighborhoods. We then merged all the spatial neighborhoods across samples and clustered them. And then we tested each cluster for basically whether it was overrepresented in cases compared to controls or vice versa, and then accounted for multiple testing. In this data set, we were unable to use existing tissue annotation methods to sort of adapt them in this way to find a case control signal. This bar chart shows the log p-value we got with each method, and the dashed line is the significance threshold. And paradoxically, the more sophisticated methods on the right actually maybe do even a little bit worse than the simple baselines on the left, which just represent each spatial neighborhood using its average expression profile or its cell type abundance profile. And as I'll get to later, this dataset does contain case control signals. We found those signals using the method we developed, which takes a bit of a different approach that I'd like to describe to you. Our method is called Variational Inference-Based Micro-Niche Analysis, or VIMA, and in developing it, there were 2 key intuitions that we tried to incorporate. First, if embeddings of spatial neighborhoods are dominated by batch effects, then case control signal might be obscured. And second, if tissue regions are forced into mutually exclusive clusters, then case control signal might also be obscured because the reality of the tissue biology might be richer than that. So here's how we operationalize these intuitions. First, we take each sample, we split it up into patches, and then we take those patches and feed them into a variational autoencoder to learn a latent representation of the patches, treating the patches as simple images, sort of pixel-level representation. This is in keeping with, I think, some themes that you guys have seen today already. But we found that when we used a vanilla variational autoencoder, the embeddings were really heavily influenced by just which sample a tissue patch came from. They were basically capturing a lot of technical variation in addition to the biological variation that we were after, even when we did aggressive batch correction upstream. To solve this, we made our autoencoder into a conditional variational autoencoder, meaning that both the encoder and decoder are given access to sample information, to sample ID. And together with the variational penalty, this basically encourages the encoder and decoder to remove technical artifacts from the latent space. This technique isn't new. It was used by Yakir Reshef and the SCVI team who used it in the single cell space, and here it made a really big difference for us. The second innovation in VIMA relates to how we do the case control association test. Here, we compute a nearest neighbor graph of the patches using the patch embeddings, but we don't then cluster the patches into what people might colloquially call niches in the field right now. Instead, we define for each patch what we call a microniche, which is composed of patches that are close to that patch in the nearest neighbor graph and ideally come from many different samples. Belonging in these microniches can be fractional, and the microniches can overlap. So the idea here is really to build a redundant and flexible representation of what's going on in each part of the graph. We then represent each sample using the abundances in that sample of all the microniches in the data set, and we find the case control signal by testing each microniche for association to case control status across samples. The result is, does this work? Yeah. An overall p-value for the presence of an association in general, and then also a measure of statistical significance at the level of each microniche that tells us whether that microniche is not significantly different between cases and controls, those are in gray, significantly enriched in cases, those are in red, and significantly enriched in controls, those are in blue. There are a lot of technical details I'm eliding here. For instance, you have to contend with the fact that there's usually tens of thousands or more of microniches. This can hurt statistical power because it makes things really high dimensional. You also have to think about how to get rigorous p-values in a way that accounts for the existence of technical replicates, which are very common in spatial data, and also the possibility of other sample level confounders that you might want to correct for. Our method does have some nice features. First, it's fast. It takes just a few hours to compute even on the large dementia data set that I described. It doesn't require cell segmentation, so it's applicable easily to many different kinds of, to really any kind of sufficiently high resolution spatial data. And it doesn't require extensive hyperparameter tuning. And you'll see in the examples I'll show here, across many different modalities, we were able to run this method with identical hyperparameters. And I won't show this here, but because we're thinking statistically, we did do simulations to make sure that the p-values this method gives are well calibrated, that 0.05 really means 0.05 and so on. And we verified that the method is well powered and that it accurately identifies the parts of each sample that contribute to the case control association or the case control signal. We applied our method to 3 data sets that I want to show you here briefly, spanning 3 really different modalities, going all the way from simple immunohistochemistry to MERFISH, which is spatial transcriptomics. The first data set is the dementia data set that I described, and this is the result of the VIMA analysis of that data set. So in this picture, what you're seeing is a UMAP where every point is a patch, and the red points are patches whose microniches are expanded in the dementia samples, and the blue points are patches whose microniches are expanded in the controls. Now, when we saw this, the first question we wondered was, what does this look like at the sample level? Like, is this a signal that is maybe significant statistically but rather weak? Is it a signal that's really driven by a small minority of the samples or something like that? And so to get at that, what we did was look at the fact that every sample in principle can contain patches of all 3 kinds, right? Every sample can have blue patches, gray patches, or red patches. And so we wondered whether the relative proportions of blue patches and red patches in each sample could, how closely they relate to the true phenotype label of that sample. So what you're seeing here is a plot in which each dot is now a sample. And this is what we're measuring. The y-axis is the fraction of patches in each sample that come from these red dots. The x-axis is the fraction of patches in each sample that come from the blue dots. And then the color of these dots is the true phenotype of those samples. And so what we see is this really is a signal that's robust and sort of exists across the data set, across all the samples, and is able to separate cases from controls quite well. What are these patches, though? To look at that, I want to show you a different view of the data set, which is a spatial view. So now you're seeing a subset of the samples, and they're sort of arrayed in space. And in each sample, I've indicated some of the cortical layers. So cortical layer 2-3, or cortical layers 2-3 are indicated in green. Cortical layer 6 is indicated in blue. And the reason I'm doing this is because when we overlay the control-associated patches onto this image, so now they're outlined in blue, what we see is that they correspond really cleanly to cortical layers 2 and 3. And this turns out to actually be a known thing. Thickness of cortical layer 3 has been associated with cognitive ability, positively associated with cognitive ability in neuroimaging studies. It's been negatively associated with dementia in single cell studies. And so, this is an example where our method is recovering something that we already know, and it's, and it makes us happy that our method is finding things that are known. Certainly in a scenario where that maybe wasn't obvious that it would work out with this modality. What about the dementia-associated patches? Well, they overlay like this. So they correspond to a subset of layer 6, not the whole thing, but a subset. And so the first question that we asked is, is this subset biologically meaningful? Is it different in any way from the rest of the cortical layer 6, or are we just sort of finding some random subset of layer 6 because we're not, because of some statistical power issue? And so to get at this, we looked at cell type abundances in basically the red outlined patches compared to the rest of layer 6. And that's what you're seeing here. So for a subset of the cells, what I'm showing you is the distribution of the abundances of these cell types across the dementia-associated layer 6 patches, that's the red histogram on the right, versus the non-dementia-associated layer 6 patches, that's the histogram on the left. And you see that some cell types really don't change much. Like L6b, this is a neuronal cell type that lives in cortical layer 6. It's basically identically distributed between what VIMA picked out as dementia-associated and the rest of layer 6. But other cell types like oligodendrocytes show really quite large differences between the patches that VIMA picked out as being typical of dementia in layer 6 and the rest of layer 6. And this is interesting because there's actually work, current work from Evan McCusker here at the Broad and others talking about the potential role of oligodendrocytes in Alzheimer's, theorizing that they might actually play a role in producing amyloid. Obviously, it's quite speculative for me to say that that's what these oligodendrocytes are doing, but nevertheless, this was an exciting example to us because it's showing a case where VIMA is finding a relatively granular association, and it's doing it where, in a place where other methods were unable to do so. The second data set is a codex data set in ulcerative colitis, and I'll speak briefly about it. The signal I'm sort of the most, excited about from this data set is a signal of TNF inhibition versus not. So TNF inhibition is the dominant treatment modality in this disease, and we were able to find basically a spatial signature that corresponds to whether patients are on a TNF inhibitor or not. And when we looked at the micro-niches driving this association, we saw a very striking pattern. So these patches here that VIMA annotates as being typical of patients who are not on a TNF inhibitor, they have this sort of stereotype pattern of clumps of B cells, that's the red staining here, surrounded by T cells here in green. These are basically lymphoid aggregates. And these lymphoid aggregates are completely absent in the cells that are annotated by VIMA as being typical of TNF inhibition. When we, again, tried to, we wanted to get a sense of how strong of an effect this was, we made the same kind of plot that I showed you in the dementia example, where each dot is a sample, and on the y-axis is the fraction of patches in each sample that come from this type of category, and on the x-axis is the fraction of patches that come from here that are typical of the TNF-naive state. And then we colored each sample by how much TNF exposure each patient had. And what we see is a really nice dose-response relationship. Basically, these two types of patches are separating patients that have never been on a TNF from patients that have been on a TNF inhibitor for a long time. So we thought this was really neat. In the rheumatoid arthritis data set, our goal was to take this immunohistochemistry, which is a relatively simple modality that's been around for decades, and to see whether our method could rediscover known disease subtypes that were only determined recently using much more advanced single-cell type technologies, and then to leverage the fact that this is a spatial modality to see if there was any extra insight that we could add. So we did see that VIMA found a very strong spatial signature of a known subtyping of rheumatoid arthritis into two disease subtypes, a stromal subtype in which fibroblast activity is high, and an immune subtype in which T cell activity is high. And I won't go into the details, but VIMA found this signal actually in an unsupervised way using essentially a type of PCA applied to the sample level representation that it builds using micro-niche abundances. And so in a sense, what this is telling us is that VIMA was able to sort of rediscover a known signal in this data set, but to do it from a modality that's been around for many, many years and that's much simpler than the single cell modalities that were used to actually uncover this distinction for the first time. And the neat thing is that because this is a spatial modality, we can actually look at heterogeneity within samples. And what's neat about this is that VIMA actually showed that although there are samples in the data set that are composed entirely of immune type patches, that's the sample here on the left, and there are samples in the data set that are composed entirely of stromal type patches, that's the example on the right. There are also many samples that are mixed that have some stromal patches and some immune patches coexisting in the same person and actually the same joint. And because this subphenotyping of stromal versus immune is thought to actually be predictive of treatment response in rheumatoid arthritis, these results have real implications for how we think about precision medicine in this disease. Like, does one patient really have one subphenotype of RA, or can there be multiple types of biology happening even within a single joint of a single person? Which is obviously not great news for precision medicine, but it's important to know if that's what's going on. So what I hope I've shown you today is that statistical case control analysis in spatial data is really a bit of a different problem that has different requirements than some of the other things that we're talking about in the spatial realm. If you want to do this, you really need a representation of spatial units that gives you consistent semantics across samples, and you need a flexible association testing strategy that can represent samples in a very rich way. And I hope I've shown you that our method VIMA enables powerful statistical case control analysis of spatial data, and that it finds new and interesting signals across a range of data modalities. So with that, I'd like to thank my collaborators, and especially Lakshay Sood and Michelle Curtis. Lakshay was part of the benchmarking effort I showed up front and led the application of VIMA to the ulcerative colitis data set, and he's actually here in the audience. As well as Michelle Curtis, who led the application of VIMA to the Alzheimer's data set, and Shoma Raychaudhuri, who's my research mentor. Feel free to check out the preprint with this QR code. The method is up on GitHub, and we'd love any feedback on it if anyone chooses to use it. Thanks very much.
I think we have a question in the back.
So yeah, I only showed a.
When you do the section samples and then you engage in the using the oligodendrocyte, why you pick the oligodendrocyte instead of some other astrocyte or microglia?
Yeah, so I guess I might be misunderstanding, but to clarify, we didn't choose oligodendrocytes when we were choosing what to input into the method. So there's no bias in that sense. I'm showing you, and what we did was compare the dementia-associated patches to the other patches across all of the cell types that were annotated by the authors of the original study. And I'm just showing you I just showed a subset for space reasons, but the oligodendrocytes are clearly, they're sort of the largest effect size, and sort of there's a statistically significant difference there. So it might be that there's other cell types that differ as well, and I'm not sure that I'm the right person to really fully understand the biology there.
Are the sample from Huntington disease or Alzheimer's disease?
No, this was Alzheimer's dementia.
Okay.
Yeah.
And actually, you picked the right cell. Because actually, last week, there's a PhD dissertation, across the street, they presented, oligodendrocyte, has some dysfunction in Huntington disease and also, to a lesser extent, in Alzheimer's.
Mm-hmm, mm-hmm.
Yeah.
Yes, it's certainly. Thanks for that info. I think maybe I'll talk to you later and see if there's a citation or something I can get for it. Yeah.
Great. Let's thank the speaker one more time.